in Spain, Associate Editor of Journal of Hepatology, co-founder, CEO, and Chief Scientific Officer at Barcelona Liver Bioservices, and is Scientific Secretary of the International Society for Hepatic Sinusoidal Research. So I welcome you, Jordi, to give this seminar today.
Thank you. Thank you very much for this kind introduction, and thank you ILBS staff, Savneet, Professor Sarin, all the excellent researchers and physicians there for the kind invitation. For me, it's an honor to be here today, and I thank you. Hopefully, we'll have the opportunity to further discuss face-to-face in the near future, but for now, we should stay online. So I will try to share my screen. Okay, can you see it?
Yes, yes.
Yeah.
Perfect. So as I said, thanks a lot. Today's talk, I would like to focus a little bit on give an update on the role of the liver vasculature, the liver vascular biology in chronic liver disease, and which have been the recent findings of our team. So these are my disclosures. Okay, so today in 40 minutes or so, I would like to go through these main areas. First, to give an introduction. I'm sure that you don't need it, but maybe it's good to recall some of the key concepts of liver vascular function and its deregulation due to chronic liver disease. Afterwards, we will talk a little bit in deep on the pathophysiology of liver vascular dysfunction in cirrhosis, and to move to novel therapeutics aimed to improve liver vascular dysfunction and chronic liver disease. So you all know how the circulation of the liver is. We certainly are aware that we have a dual blood inflow coming from the hepatic artery and the portal vein that drains the blood through these microvessels or this microvasculature of the liver, which is called the hepatic sinusoid, which is totally unique in the body. And this is composed mainly, the liver sinusoid is mainly composed by three main cell types. First, we have our Professor Dr. Kaur and myself, our favorite cell type, which are liver sinusoidal endothelial cells, which we know that they are discontinuous, they present fenestra, and they lack of basal membrane, which facilitates the communication between the bloodstream and the parenchyma. And they play key roles in liver homeostasis, in the control of inflammation, clearance of toxicants, and also the regulation of vascular tone. On the other side, we have hepatic stellate cells, which are the pericytes, the contractile cells of the liver. They have, as I said, contractile properties, and they are the main storage of vitamin A in healthy situations. Finally, we have resident macrophages, which are called Kaffer cells, and they play a key role in defense, in inflammation, and in tissue remodeling. We know that these cells that they communicate very well during normal, during a healthy status, we know that these cells present, as I said, fenestra, vasodilatation in endothelial cells. Hepatocytes are able, also the parenchymal cells, obviously they communicate with each other and they play key roles in synthesis of proteins or metabolites and detoxification. Stellate cells are quiescent with these lipid droplets full of vitamin A, and Kaffer cells are anti-inflammatory. Nevertheless, it's quite well known that in response to an injury, and this can be acute or persistent injury, the first cells that sense this damage and react to this damage are endothelial cells that become capillarized, dysfunctional, and also hepatocytes that become necroapoptotic. And at the end, when we have the situation of advanced or chronic liver disease, we have a complete deregulation of all cells of the hepatic sinusoid. Liver sinusoidal cells are capillarized, activated Kaffer cells, activated stellate cells that, as you very well know, synthesize and release extracellular matrix components, and obviously necroapoptotic hepatocytes. It's important to have an overview of chronic liver disease progression And we can separate this progression of cirrhosis in two main parts. First, we have this epithelial injury and the associated inflammatory response. And as a secondary, we have hepatic stellate cell activation and the perpetuation of disease. So, as I said, we have this first insult that mainly targets hepatocytes. These hepatocytes become injured, apoptotic, and necrotic. We also know that damage has a direct effect on the phenotype of liver sinusoidal endothelial cells. Hepatocytes start to release different pro-inflammatory cytokines, growth factors, apoptotic bodies, extracellular vesicles, oxidative stress mediators, etc. This is also complemented with the release or the contributions of different myeloid cells, even from platelets, that generate this mixture of different pro-inflammatory molecules. This activates Kupffer cells, but also recruit myeloid cells and macrophages towards the liver. And we also know that these endothelial cells, after the damage, they lose the fenestra and they acquire basal membrane. They also, we know that from the beginning, now we have this in a healthy situation, quiescent hepatic stellate cells, but obviously the release of all these pro-inflammatory mediators also from macrophages, both resident or recruited, and also different mediators, vasoconstrictors, microvasicles, pro-inflammatory mediators from endothelial cells, altogether, they contribute to the activation of hepatic stellate cells, which become procontractile, which increase their migration, they lose the lipid droplets, and they synthesize and release extracellular matrix components. Also, they have an autocrine activation pathway mainly mediated by TGF-beta. So in front of this global deregulation during the progression of chronic liver disease, we have major consequences. One is the increment in fibrosis. We know that the cirrhotic liver has elevation, a high degree of fibrotic tissue. Also, the stiffness of the liver, which, as I will talk in a few minutes, further perpetuates the activation and the regulation of all hepatic cell types. And on the other side, we have an increase in the intrahepatic vascular resistance that I will talk a little bit now. And this leads to an increment in portal pressure and the development of portal hypertension syndrome. It's important that these are the main pathophysiological targets in patients with chronic liver disease. If we are able to develop treatments to improve the vascular resistance, we will probably improve the prognosis of these patients. The same if we develop novel antifibrotic agents or novel agents to inhibit the stiffness-mediated molecular pathway. So if we focus a little bit more on detail on portal hypertension, all of you know that portal hypertension is a clinical syndrome which is very frequent in patients with chronic liver disease. And this is characterized by a pathological increase in the portal pressure gradient above the normal value, which is around 5 millimeters of mercury. And portal hypertension has key clinical consequences in patients. We know that patients, they develop or they suffer from the bleeding from gastroesophageal varices. They develop ascites or hepatorenal syndrome, gastropathy, hepatopulmonary syndrome, and hepatic encephalopathy, among others. And all this leads to a very bad prognosis in patients with clinically significant portal hypertension. And how should we understand the pathophysiology of portal hypertension? I think we should look at portal hypertension from a global point of view. So we know that the initial factor, as I described, is the deregulation in all cell types of the liver. Hepatocytes become necropoptotic, endothelial cells capillarize, hepatic stellate cells activate, and the immune cells also become activated and recruited. And all this together leads what we call the development of hepatic microcirculatory dysfunction. This hepatic microcirculatory dysfunction has two key consequences. One is the development of several architectural disturbances in the liver, which include the development of fibrosis, vascular remodeling, generation of regenerative nodules, etc. And also we have an increment in the hepatic vascular tone that is characterized by a higher release and synthesis of vasoconstrictors, reduced number of vasodilators, and also sinusoidal hypercontraction. Altogether, this leads to an elevation in the hepatic vascular tone, in the hepatic vascular resistance, which is the primary factor in the development of portal hypertension. Obviously, and as consequence of this increment in the portal pressure, in the sinusoidal portal pressure, there is a compensatory mechanism that leads to the splanchnic vascular cells to generate vasodilators, but this at the end contributes to the splanchnic vasodilatation, an increase in the portal blood inflow, and this increase in the portal blood inflow further perpetuates and aggravates the portal hypertension syndrome. So if we focus on the intrahepatic part, on the events that happen inside the cirrhotic liver from a vascular point of view, we know that this is the main or the primary events contributing or promoting portal hypertension. But also the architectural disturbances play a key role. So we know that as much fiber, as higher is the percentage of fibrosis of septa in cirrhotic livers in human cirrhosis, we know that HVPG or the gradient in the portal pressure is also increased. So as high the fibrosis, higher will be the portal pressure of these patients. And this is important because we can also validate the same results that are observed in different cohorts of patients with chronic liver disease. We can observe the same in preclinical models of cirrhosis. So controlled healthy animals have a controlled portal pressure, but during the progression of the disease and the deposition of extracellular matrix, this is a early cirrhosis and this is advanced cirrhosis. You can see how portal pressure increase in association or in proportion of the increase of extracellular matrix. And this fibrosis, also angiogenesis, we know that cause a progressive distortion of the sinusoidal microcirculation, of the liver microcirculation. They promote sinusoidal remodeling. And as I said, this leads to an increase in the intrahepatic vascular resistance, so contributing to portal hypertension. And also we know, and we will talk further, that capillarization of these sinusoids, the deposition of this extracellular matrix that impedes the correct communication between the hepatocytes and the bloodstream, this contributes to liver failure. Here you can see some beautiful images from our team where you can see how beauty is the sinusoidal circulation in healthy livers. You can see a perfect network of microvessels from the portal area to the central area. And here, the same with lower magnification, and how different it looks when we analyze at the same magnification livers from cirrhotic animals or cirrhotic patients. You can envision how difficult it will be for the blood flow to go through the cirrhotic liver with these architectural disturbances in the parenchyma. And in this point, I would like to introduce the stiffness. So up to now, liver stiffness has been understood as a consequence of chronic liver disease, as a consequence of this fibrosis and this vascular remodeling, and therefore contributing to or as a surrogate marker of portal hypertension. Nevertheless, for the last years, we have tried to think on or try to change the dogma and evaluate stiffness or consider stiffness not only as a consequence, but also as a cause of chronic liver disease. And indeed, we have evaluated how or whether liver stiffness may further contribute to the development or the aggravation of hepatic microcirculatory dysfunction in the cirrhotic liver. So for this, in our lab, mainly Sergi Guichet and Martí Ortega from our lab in Barcelona and in Bern, they have been working with different in vitro models of stiffness, on how to study stiffness in vitro. And we can do that with different biocompatible gels, where we can modulate or we can generate the gels with different stiffness. For example, in the experiments that I will summarize now, we use healthy stiffness as a 0.5 kilopascal, fibrotic stiffness as 13.5 kilopascal, and 30 kilopascal as a mimic of chronic liver disease stiffness. And when we generate these biocompatible gels, we can work with them in vitro, we can plate cells in vitro, and we can test how these cells behave, how is the phenotype of these cells when we cultured them at different stiffness up to 72 hours, for example. So it's interesting, and as a summary of this very recent paper that is now available in JF Reports, that when we check how is the phenotype on how stiffness modulate the phenotype of healthy cells, so healthy hepatocytes, healthy hepatic stellate cells, and healthy LSEC that are cultured in different stiffnesses, when we culture them in high stiffness, we know and we observe that they very change, they modify their phenotype. Hepatocytes become dysfunctional, hepatic stellate cells activate, and endothelial cells become capillarized. It is very true that the observations of dysfunctional hepatocytes and activated stellate cells due to high stiffness was previously characterized. Nevertheless, we were the first, in these experiments, were the first analyzing and discovering how endothelial cells behave under high stiffness. And indeed, healthy endothelial cells culture under high stiffness, they show, as you can see, a marked deregulation in their phenotype. So, for example, as a surrogate marker of nitric oxide of endothelial status of endothelial function, the analysis of nitric oxide, you can see here in green some representative images, and here you can see the quantification. The release of nitric oxide of healthy cells culture in high stiffness was significantly lower than those cultured in healthy stiffness, so suggesting a spontaneous capillarization just because of the high stiffness of the substrate where the cells were cultured on. And similarly, when we analyze fenestrations as a gold standard method to analyze or to understand the phenotype of endothelial cells, you can see how they reduce the fenestration, both the porosity and the frequency of fenestra in endothelial cells when we culture them in high stiffness. So indeed, suggesting that these endothelial cells become capillarized in high stiffness. Interestingly, when we analyze which was the role of stiffness on the phenotype of cirrhotic cells, so in cells isolated from cirrhotic livers, we observe the contrary. We observe how these cells, when they are cultured in low stiffness, so in healthy stiffness, they recover the functionality. Hepatocytes recover functionality, hepatic stellate cells deactivate, and endothelial cells recover their functionality. And this, it can be observed, for example, as a matter of example, hepatocytes, cirrhotic hepatocyte culture in lower stiffness release higher amount of proteins, for example, urea and albumin. We observe this at the protein level, also at the mRNA level, so suggesting an improvement in their functionality of hepatocytes. Also, hepatic stellate cells become deactivated. You can see very clearly the expression of alpha-SMA both at the mRNA and also at the protein level, how is decreased just because we are culturing these activated stellate cells on healthy stiffnesses. And similarly, liver sinusoidal endothelial cells become functional, become decapillarized. So we can see that they release higher amounts of nitric oxide in lower stiffnesses, 0.5 kilopascal. And this is accompanied by a trend in the increase in fenestration. Please consider that here the only change between conditions is the stiffness of the substrate. So just improving stiffness, we are able to markedly improve the phenotype of cirrhotic liver cells. And how? No? So we can conclude that substrate stiffness per se modulate liver cells phenotype both during the progression of the disease and also, presumably, during the regression of the disease, no, in low stiffnesses. And how this happens, no? So to understand the mechanism, we envision this paradigm, no? In a normal situation, we know that cells, they constantly test which is the stiffness of the substrate where they live. So they sense the stiffness, and this is done through the cytoskeletal union, the cytoskeletal link between the extracellular domains and the nucleus. So cytoskeletal contraction would happen only if the extracellular matrix is rigid. So in a soft matrix, so as we can envision or we can think on a healthy liver, this cell become round, become as a circle, no, as a spheric, and the nucleus also becomes spheric because there is no contraction of the cytoskeletal forces. Nevertheless, in a rigid matrix, you can see how the cell becomes distortion, and importantly, there is also a distortion in the nucleus due to this contraction in the cytoskeletal proteins that are anchoring the extracellular domains and the nuclear domains. And this for us was the key point, or we hypothesized that this was the key point to modify the phenotype of the cells and promote the activation of these cells. And indeed, when we analyze the nucleus of hepatocytes, stellate cells, and endothelial cells, cultured at low stiffness and high stiffness, and we analyze how was the shape of the nucleus. You can see here a reconstruction in the shape of the nucleus. So you can see how this is totally or mostly round in situation of low stiffness. Nevertheless, in high stiffness, the nuclear shape changes and becomes similar to a coin. So you can see that there is a high deformation of the nucleus. Importantly, when we can analyze or we can quantify the deformation of the nucleus, you can see here the quantification. This is the deformation at the different deformation, let's say, stages. And you can see that at high stiffnesses, most of the cells are highly deformed. And this is similar when we perform the global quantification of all cells. So these cells in soft stiffness have a low deformation degree, high deformation degree in high stiffness. Importantly, this is also true when it is analyzed in tissues from cirrhotic patients. So here you can see representative images of hepatic cells nucleus from healthy patients, patients with chronic liver disease due to ethanol consumption, and cirrhotic patients due to NASH. You can see also that in this situation, and these are tissues, as I said, from human beings, so you can see how the cirrhotic tissues, cirrhotic cells, also present a deformation in the nucleus. And importantly, this is a campaign. You can read the full paper if you wish to see how this phenotype, so this nuclear deformation is associated with a phenotype change, a dysfunction in the phenotype. And importantly, this can be targeted by different strategies targeting the cytoskeletal proteins, for example, cytoskeletal disruptors or dominant negatives of proteins of the nuclear envelope. But if we use these cytoskeletal disruptors, at least in vitro, and cells are cultured in rigid matrices, so in high stiffness, they recover the spherical nucleus, and importantly, they recover the healthy phenotype. So suggesting that indeed, if we are able to develop novel treatments that are functional or that are inhibiting how the cells sense the stiffness of the cirrhotic tissue, we may improve the phenotype of the cells without changing the stiffness, but changing how the cells, they sense and they react to the rigidity of the liver tissue. On the other side, once we have talked about the architectural disturbances, we know that on the other side, we have the increment in the hepatic vascular tone contributing to the hepatic vascular resistance. And this, as you should know, this was discovered so many years ago by Bathal and Grossman in this pioneering study in 1985, where you can see these are very easy experiments using the ex vivo perfusion system to perfuse the livers of normal healthy rats or cirrhotic rats. And these livers under perfusion, so what they did in this pioneering study was to add to the reservoir nitroprusiate, which is a donor of nitric oxide, or prostaglandin E1, which is a vasodilator, a prostaglandin which with vasodilatory properties. So you can see that control livers with a normal, healthy hepatic vascular resistance, they do not modify the portal pressure in response to vasodilators. Nevertheless, you can see how immediately after adding nitric oxide or prostaglandin E1, cirrhotic livers, they relax and they vasodilatate significantly. So this was the first time that it was suggested that the increment in the portal pressure was not just due to the distortion in the parenchyma, but also to a dysfunctionality of the vascular cells. Those students that are watching this seminar should read this paper. I know that this paper is not available online, but you can go to a library and check for Journal of Hepatology paper, no, in the real volume, 1985, and please read the paper. And why and how we can understand the dynamic component of the hepatic vascular resistance. To do that, we should go to the Poisson law that says that the resistance of a vessel, the resistance indeed of any hydrodynamic system, is inversely proportional to the radius of this vessel. No? And you can see if we move and we observe how is the sinusoid, and we consider this Poisson law, resistance is inversional proportion to the radius. So here we have the radius. So if we are able to develop treatments to increase a little bit the radius, a little bit, to dilate a little bit the radius of all the sinusoids of the liver, we will able, because this is at the fourth, no, we were able to reduce the resistance significantly. So we should develop treatments aimed at this, at increasing the radius, vasodilate the sinusoid, and this in consequence will reduce the resistance. And how can we do that? So we should take in consideration that we have different intrahepatic contractile elements, mainly hepatic stellate cells, at least in the sinusoid. And also we have different cells that regulate the contraction of these stellate cells, mainly endothelial cells, but also Kupffer cells. And this is done by paracrine interactions between non-parenchymal cells, endothelial cells, Kupffer cells, also systemic vasoactive molecules that reach the liver. And you can see here a summary of some of these paracrine factors that ultimately lead to the contraction of hepatic stellate cells, norepinephrine, thromboxane, urotensin, leukotrienes, endothelin, etc., etc. And importantly, very few amount of vasodilators are found in the cirrhotic circulation, in the cirrhotic liver. Very low nitric oxide by different mechanisms that I will not explain today, but we have low levels of nitric oxide, low levels of H2S, no, sulfhydride, carbon monoxide maybe, etc. So we have low levels of vasodilators and high levels of vasoconstrictors. So further promoting the constriction of a stellate cell and the increment in the hepatic vascular tone. So to summarize how we, considering this knowledge on the pathophysiology, how we can target the hepatic vascular resistance with the aim to promote a reduction in the portal pressure, we can here summarize, this is a summary of different pathways that lead to vasodilatation, mainly nitric oxide. So we know that the endothelial nitric oxide synthase use L-arginine to produce citrulline, but nitric oxide importantly, and this nitric oxide will activate the guanylase cyclase to generate cyclic GMP, and this is the molecule that will promote vasodilatation. Nevertheless, we know that there are inhibitors, for example, phosphodiesterase inhibitors that promote the degradation of cyclic GMP. So this would be one of the main vasodilatation pathways, but we also have several, as I said, vasoconstrictor pathways, urotensin, endothelin, different eicosanoids, angiotensin II, etc. So during the last decades, and this is work from our team, but also from the, for example, ILBS team and other teams in the world, we have developed or we have tested different potential therapeutics targeting these specific pathways that we know that are regulated in the cirrhotic microcirculation, in the cirrhotic liver microcirculation. So for example, targeting nitric oxide synthase, we have FXR agonists, maybe statins, one of the most promising together with OCA, they promote the expression and also the activity of nitric oxide, different cofactors of nitric oxide, tetrahydrobiopterin, folates, etc. NO donors have been tested. Let's say systemic donors, but also with particles, different activators of the 1-11 cyclase, inhibitors of the PDE, no, phosphodiesterase, and also different teams, including ours, we have tested the potential of targeting all these vasoconstrictive pathways, no? Urotensin inhibitors, endothelin inhibitors, eicosanoid inhibitors, the inhibitors of the renin-angiotensin system, JUNC, or Rho kinase. You can see here, this is a beautiful summary that we did together with Sergi Guichet and Marina Vilaseca, how those that are involved have been tested at the bedside with positive or not positive results. But you can see that some of them, the preclinical work that we develop in the lab, in some cases is moved to the clinical scenario. And to go a little bit on detail, and probably, as I said, one of the most promising are statins, and in particular simvastatin. We have been working, and in this review that we just published in GAD, we have been trying to concise all the work that we and others have been doing, both at the preclinical and clinical scenario, working with the statins. And we have evidence that the statins may be protective, both in the acute liver injury situation. We know that mainly targeting the KLF2 pathway. We know, well, you can see that different cell types are targeted, which are the underlying mechanisms, which are the pathologic events that are improved. All this is coming from preclinical models. You can see a summary of preclinical models. But interestingly, also there are some clinical evidence, observational studies for acute liver injury. In the case of chronic liver disease, of cirrhosis, mostly this work has been done by the group in Bonn, now in Frankfurt, and our team in Barcelona. So you can see that this is through a KLF2 pathway, probably also through Rho kinase. And you can see that this has been tested in different preclinical models, and importantly, in observational studies, in proof of concept randomized clinical trials, and also in clinical trials with very clear endpoints. And interestingly, and this Dr. Tripathi, which I'm sure will be listening this seminar, also contributed to the understanding on how statins may improve ACLF. And we developed a preclinical model of ACLF, administering LPS to rats with chronic liver disease. We test which were the different mechanisms and the benefits of administering simvastatin in this very advanced situation of chronic liver disease. And interestingly, there is an ongoing phase 3 randomized clinical trial also analyzing the effects of simvastatin in this clinical situation. And to summarize the underlying mechanisms of statins improving the vasculature of chronic liver disease, we know that statins, by inhibiting the HMG-CoA, leads to a decrease in the isoprenylation of different small GTPases, mainly RAG1 and Rho-A, and this leads to a reduction or an inhibition of their downstream pathways, leading to an increment in KLF2, an increment in nitric oxide, reduction in oxidative stress, reduction in the contraction of cells, etc. So we know that in endothelial cells has vasodilatory, anti-inflammatory, antithrombotic, and antioxidant effects, and in the stellate cells, the statins have vasodilatory, antifibrogenic, and antiproliferative effects. Importantly, also we can promote an improvement in the intrahepatic vascular resistance, targeting the pathophysiology, the big pathophysiological events occurring during chronic liver disease, which can be summarized as an increment in oxidative stress, an increment in cell death, and an increment in inflammation. This is like the vicious circle of chronic liver disease from a vascular biology point of view. And this ultimately leads, as we are discussing, on fibrosis. Also, different preclinical studies and also clinical studies involved, you can see different therapeutic agents targeting antioxidants, resveratrol, dark chocolate, vitamin C, others, anti-apoptotic drugs. It is true that both emricasan and selonsertib had very nice or very promising effects at the preclinical scenario that were not validated in the clinical setting. Anti-inflammatory, obviously we can modulate this inflammation and chronic liver disease using microbiota modulators, different anti-diabetics, liraglutide, metformin, and also very interesting, and you at Delhi also are working very hard on this topic, if we can use regenerative medicine to improve chronic liver disease and portal hypertension. So we have the different examples using amnion-derived stem cells, bone marrow stem cells, microvesicles isolated from stem cells, or also liver cells progenitors. This is due to the time, I cannot explain the whole study, but I recommend you to read this paper that has been just recently published in Liver International. This has been done in collaboration with ISMET in Palermo, Italy, where Giada Petrosian and Abel Fernandez, they have tested the effects of amnion-derived stem cells, that human origin, their effects as a paracrine release vasoprotective cells, both in vitro, and they demonstrate how improve the phenotype of stellate cells and hepatic stellate cells in vitro. And also when they are injected to cirrhotic animals, they improve the vascular function, portal hypertension, and the phenotype of these main cells playing a role in portal hypertension. So I strongly recommend you to read the paper. And before wrapping up, I would like to acknowledge, obviously, all my team, both in Barcelona and in Bern. They work very hard, and obviously I could not give this talk without their work and their support. Mainly, well, all of them are contributors, collaborations that we have around the globe. I really look forward to start collaborating with ILBS in the near future, and obviously the funding agencies that support our work and our especially during these difficult days in I think worldwide. So to wrap up, we know that chronic liver disease leads to the development of liver vascular dysfunction, and this liver vascular dysfunction is due to the deregulation in all cell types: endothelial cells, stellate cells, Kupffer cells, and recruited macrophages, and obviously dysfunctional hepatocytes. We also know that chronic liver disease is a very complex disease where, let's say, four main pillars: inflammation, oxidative stress, the development of fibrosis, and obviously vascular dysfunction. Vascular dysfunction, oh my God. Vascular dysfunction play a key role. Inflammation, we know hepatocytes, macrophages, LSEC all contribute to inflammation. In oxidative stress, all the cells release oxidative stress mediators. Fibrosis, obviously stellate cells are the star because they release the extracellular matrix components, but it's very important the interactions with other cells, the decrease of metalloproteinases, obviously, as I explained to you today, the increasing stiffness playing a key role in the progression of the disease. And vascular dysfunction, it is indeed a key component because leads to the development of portal hypertension, and this, as you very well know, has detrimental consequences for patients with chronic liver disease. And finally, we and others are working hard for the development of new therapeutics for liver vascular dysfunction. And here, this is a nice cartoon that we developed in a late review that is now available. You can see how we can improve stellate cells by several mechanisms targeting KLF2, enos, nitric oxide, also targeting different vasoconstrictors, improving macrophages, inhibiting different vasoconstrictors, mostly done or happening in the stellate cells, but also, as I said, modulating gut microbiota, inhibiting the apoptosis of stellate cells, of hepatocytes, and hopefully we will be able to develop novel agents to remodel and to degrade the matrix in the situation of chronic liver disease. And with this, I finalize. Again, I would like to thank the kind invitation to be part of this ILBS seminar series, and I will be very happy to take any question. Thank you.
Questions? Great, thank you so much. Okay, so have you checked the effect of statins on the mechanobiology or stiffness? I mean, apart from KLF2, any?
Yeah, this is an interesting question. So we have tested two different drugs. How is their efficacy under different stiffnesses? So we observed that statins were as good as expected in both high or low stiffness. But for example, liraglutide there was a difference in their efficacy deactivating the stellate cells depending on the stiffness. So this leads to think on the possibility that in addition to the therapeutic windows that we know that exist during the progression of the disease, for example, for using beta blockers, we may also envision that there is also a therapeutic window for each target, for each therapeutic targeting the vasculature, depending on the stiffness. Obviously, this has to be assessed, and this has to be assessed in a large number, using a large number of different drugs. But at least with liraglutide, this was true. It was effective at a certain stiffness, and it was less effective at a different stiffness. So I think we should work on that and check if we can define a therapeutic window that depends on stiffness.
Jody, I have a question. You mentioned that your study is focused around the statins to treat this class of fibrotic class of patients, and this being a multifactorial disease. So, and you have shown so many pathways getting involved, and also individual inhibitors. Did you try combination of these?
With other drugs, you mean? Or with which drugs?
Say anti-apoptotic with anti-diabetic or anti-inflammatory and so on.
We haven't tried that. It's true that is. So I think the good point on the statins is that they are dyspleiotropic, that they will have higher chances of success probably than using a specific molecule targeting a specific pathway that may be inhibited, but probably other pathways play a role also, and they overcome the inhibition that you can obtain with a single molecule. So that's why I think that, for example, obeticholic acid that is acting on also a transcription factor, or statins that are acting on different pleiotropic pathways, I think they have higher chances of success than using specific agents. In the past, our team tested, for example, using sorafenib, which is an anti-angiogenic, with beta blocker. This was developed by Mercedes Fernandez in our team and Jaime, and they saw some kind of synergistic effect. And I can tell you that I have been in contact with a paper that is now under revision, where there is a combination of a statin with another drug, and also they observe some kind of synergistic effect, but I would say that lower than expected. Probably, as I said, because statins are quite pleiotropic, and probably it would make more sense to combine statins with beta blockers because they act on the two main underlying causes of portal hypertension. Statins mainly acting intrahepatically, beta blockers mainly acting on the splanchnic circulation, and this may have a greater effect than combining with other drugs targeting also the intrahepatic circulation.
Okay, now we have questions from audience here. Yeah, please.
Yeah, so one question coming from Dr. Gayatri. Besides the cytoplasmic cytoskeleton, is there a change in nuclear matrix proteins?
Very, very interesting point. Yes, there is a change, and this is what we are studying now. So we are analyzing now how these cells change. in response to stiffness, we observe that there is a gradient in different proteins, but we are now studying which proteins and which role they may play in response to stiffness. But we have no data, not yet. We are studying that.
Okay, so another question from Dr. Jaswinder. Role of calcium channel blocker inhibition in fibrosis modulation or activation of stellate cells. I mean, pan calcium modulation versus some site-specific
I don't recall any. Maybe I'm not. You know that I'm not a stellate cell man. I'm an endothelial man. No? So, but if I remember well, some studies from Ramon Bataller, from David Brenner, 10 or 15 years ago, no, they were analyzing different calcium channels inhibitors. We did some studies on potassium channels, but I don't recall. Maybe I'm not right, but I don't recall the comparison between pan calcium or a specific calcium modulation. So I'm sorry, but I cannot understand. I cannot reply.
Okay. So
Jordi, I think the 3D simulation of the nucleus that you showed, was it based on morphometry or some other parameters?
Correct. It was morphometry. So this was elegantly done by my fellows, that they can explain much better than me how it was done. But this was some kind of using confocal microscopy. They took several images at different zeta axes of the nucleus, and afterwards they reconstructed the whole nucleus of the cell. Yeah.
Okay.
So I just wanted to ask that there was a recent paper which was talking about etiology-specific changes in fibrosis. So do you think that, I mean, all kinds of fibrosis, irrespective of etiology, they culminate into a similar kind of process, or there might be some changes because of the advent of these single cell sequencing kind of sophisticated techniques? People are now saying that it's an etiology specific mechanism which might be operating and not same for all fibrosis. So
I totally agree with you, Savneet. So I think the mechanisms happening during the progression may be different depending on the etiology. For example, we have unpublished data comparing the transcriptome of endothelial cells, from different preclinical models at different ages, young versus age, and also human etiologies. And we observe that they are different in a certain percentage of genes and pathways. They are different, but also they share a lot of common pathways that are deregulated. So I would suggest that I would say that maybe we have a mixture of both divergent pathways that are specific of the etiology. For sure, a cholestatic injury is very different from a NASH injury. It's the origin is different, and the paracrine interaction between cells will be different, and the cellular deregulations will be different. But I'm sure that they share some similar pathways and deregulations that are interesting, because probably if we are able to develop and to validate treatments for those shared pathways, probably they will be given, or will be beneficial for a larger amount of patients.
Okay, thank you. So
okay, I think thank you, Jordi, for being with us this afternoon, and it was really a great talk. We learned a lot about the mechano aspect of the liver disease and being multifactorial. What all factors could be crucial, what all pathways could be crucial. So it was a good learning exercise for us, and definitely, as pointed out by you, our strength is regenerative medicine. So we'll try to look at this problem from that angle. Thank you so much for being with
Thank
us,
you.
and we look
Thank you.
forward to some collaboration in future. Okay.
It has
Thank
been
you.
a
Great.
pleasure. Thank you. Thank you so much.
Take care. Stay safe.
Take care. Bye-bye.
Ciao.
Okay.